I am going to be a senior at Upper Arlington High School in Ohio. The main focus of my research is to try to discover or identify a drug molecule that will help stop nicotinic addiction. And the current treatment options that are on the market include nicotine replacement therapy, varenicline, as well as bupropion. Now this whole nicotinic addiction problem is due to nicotine, which is found in all tobacco products, and it facilitates the addiction process by interacting with nicotinic acetylcholine receptors, which are found in the central nervous system. So these current treatment options, unfortunately, all of them have very low success rates. For instance, nicotine replacement therapy and bupropion are about 15%, and varenicline about 25%. And varenicline, which has the greatest success rate, actually has some serious side effects, which obviously we're trying to minimize when administrating any drug. The current drugs on the market which target the orthosteric site, which I mentioned earlier is the site where nicotine binds to the receptor, you target the allosteric site, which is a site that is unique specifically to the receptor that you are targeting, which in this case, I'll be targeting the alpha-4 beta-2 nicotinic receptor subtype. And basically when the drug molecule will bind to the receptor at this allosteric site, what you get is a conformational change, essentially a dimmer switch, I guess you can say, that closes that central pore that I told you about earlier. And this stops the flow of calcium ions into the cell, and this stops the eventual downstream release of dopamine. So basically, the individual doesn't receive the high that they would usually from smoking. Now, the upside to allosteric modulation is that you can discover a more selective as well as potent molecule, because, as I said, this site is unique specifically for the alpha-4 beta-2 receptor. So if you are able to design this molecule, you can, it's much easier to design it for this, specifically the alpha-4 beta-2 receptor. So the first step in my research was to construct a Pharmacophore model. Basically, a Pharmacophore model highlights the essential structural features of that are necessary for activity. Now, here you can see this is constructed from 5 molecules that I'd identified from my previous year's research to have some level of potency and selectivity, and basically using a computer program, I was able to overlay these 5 molecules and come up with this scaffold, I guess, that highlights, as I said, these essential structural features for activity. So, using this Pharmacophore model, I carried out the virtual screening process, essentially screening this Pharmacophore, using a computer against a 10,000 molecule library. And from this 10,000 molecule library, I was able to get 150 hits, from which I chose the top 11 for further testing. The next step was to actually test these 11 molecules in vitro, and for the purpose of this, I used human embryonic kidney cells, which are transfected with human nicotinic receptors. And the 2 receptor subtypes that I chose for my research were human alpha-4 beta-2 and alpha-3 beta-4. The subtypes because, the alpha-4 beta-2 are the receptors that I'm targeting, and alpha-3 beta-4 are the most prevalent nicotinic receptors in the autonomic nervous system, and they control a lot of involuntary emotions, such as breathing or muscle contractions. And so you're pretty much trying to avoid those at all costs to minimize any kind of side effects that could hurt the patient. As a kind of primary screening method, I tested these 11 molecules and was able to acquire this data. And my measured variable here is fluorescence intensity, and this is basically a direct measure of the calcium concentration within the cell. What you're looking for at the alpha-4 beta-2, which are the black bars, is a lower fluorescence intensity, and you're also looking for a greater difference between the 2 bars in order to look for selectivity, because obviously we're targeting the alpha-4 beta-2 over alpha-3 beta-4. And The standard which I used, or the agonist in place of nicotine, is epibadidine. So obviously 100% fluorescence intensity is basically no activity, which is what we're looking for alpha 3 beta 4. So the next step here was to construct dose response curves using these 11 molecules. And essentially what this does is helps to determine IC50 values, and IC50 value is the concentration at which you see 50% inhibition. This was the most potent and selective molecule that I was able to find. As you can see, the IC50 for alpha 4 beta 2 is 6.4 micromolar, while the IC50 for alpha 3 beta 4 is 81 micromolar, which is greater than a 10-fold difference, which is huge. And the other two which I found were hit number 6 as well as hit number 9. And if you look at the structures of hit number 9 and hit number 4, they're quite similar, while hit number 6 is more straight chain. So the last step of my research was to construct contour maps. And contour maps, basically, you try to determine a hypothetical structure of the ligand binding domain. And unfortunately, the 3D crystal structure of these nicotinic receptors haven't been determined yet. So to be able to design a molecule that'll better interact with these receptors, it would be better if we knew what they were interacting with. So if we were able to determine the structure of the binding domain, you could more easily design a molecule that fit. It's like a lock and a key. So basically, this is the colored areas are the contour map. So basically, we get a further insight into the ligand binding domain, which is pretty important, like I said, if we want to optimize the activity of the drug molecule. So basically, what we can conclude from this is, that we were able to generate a pharmacophore model, which was used to identify a novel antagonist. Additionally, we were able to find a drug molecule with a potency and selectivity in the micromolar range, which is a huge feat in itself. A 10-fold difference was seen in potency between in hit number 4, which was again a huge feat. And finally, as I said, we were able to gain further insight into the structure of the binding domain, which leads me to my ongoing work, which would be to use the contour maps to optimize the activity, to increase potency and selectivity of the molecules. And I'd just like to thank my chemistry teacher, Mrs. Brennan, Professor McKay, as well as the PhD student that I worked with, Brandon Henderson, my friends and family who are also here, as well as the NIH for their support.
Jana, I want to congratulate you again. I mean, it's a wonderful study that you're doing in and of itself, but I'm also very impressed about your resourcefulness, because what you've been able to do is get access to the computational infrastructure, and to the molecule databases that allow you to test them. So that's pretty impressive that you already got your hands into that. Now, my question, too, because you are so resourceful, so sort of, have you thought of giving these molecules to rats and see does it have any behavioral effects?
Well, that would be the eventual goal in the long run. I don't think we're ready for that as of yet. But once we've kind of looked further into pharmacokinetics and things like that of the drug molecules and have reached a certain level of potency that we feel is desirable, we can move into the preclinical trials. So yeah, not as of yet, but eventually in the long run we can deal with that.
So at this point you are not considering issues like lipophilicity, whether it's going to cross the blood-brain barrier or not?
Right. That's not our main issue right now. We're more on the drug discovery process, but once we've identified a molecule that we think has the desired characteristics, then we'll further look into the things like what was the term?
Blood-brain barrier.
Whether it's able to cross the blood-brain barrier, the bioavailability, things like that.